The key part of this is that it's a data management system, so we're focusing on a lot of the data associated with photo ID. So as you all sort of realize, photo ID is hugely time consuming and visually fatiguing, and we wish we just really didn't have to do it at all. But it's subject to human error, and when we're going between different photo ID projects, it also, taking that cross comparison takes a huge amount of time as well. So to help alleviate some of the time consuming part, there's been a number of systems developed in the last 15 years. And this is an example of the FinScan that was developed in the late 90s, where we tried to use, look at dorsal fins and identify what the notches are and come up with different algorithms to look at that. But these have only focused on the matching part of photo ID and are usually very species specific. And they're not completely automated, so that's why they're computer assisted. They basically require the user. And in some ways, like FinScan or Darwin and that, they could also increase potential time to process photo ID because you need to play with a lot of the image segmentation because they're not perfect. So there's a lot of time required as a part of that. But there's so much other data associated with photo ID. Not only the life history parameters associated with the individuals, but you also have a lot of sighting information that goes into group size, spatial patterns, etc. And a lot of this has not been the focus of different software. And because those other softwares are so species specific, there's so many other different species that there's no software available for. So we tried to develop an overall comprehensive approach to store, visualize, manage, and analyze your photo ID data. And we realized that there are really no standards in the field, and what John Moran presented yesterday and how he processes photo ID is totally different than another research lab that may process photo ID. They do image quality in one way, we may do it another way. So there's different methods in photo ID how to process. So we really tried to make this a dynamic system to change over time, and also whatever your research project's needs are. And we realized that we're getting more and more long-term data sets, and so this really requires collaboration between different research groups. And so we really need to try to standardize databases, because doing those comparisons between different databases from different labs is hugely time-consuming as well. So as we were creating this, we also realized, we don't want to reinvent the wheel. So we tried to be inclusive as much as possible and not exclusive of other tools. So we tried that as much as possible. And we realized, for one species, there can be multiple catalogs. For example, with gray whales, just on the population of western gray whales, we have 4 different catalogs that we all have to interact with. And we all have different identification numbers. So we can interact with them, and also for the eastern gray whales, there's a number of different catalogs. So if we really want to understand the species and the individuals, where they occurred throughout geographic range and throughout their life history, we really need to take use of all those different information. So we need to extend collaboration. But even with one single study area, for example, in Kaikoura, I can go out in one day and identify 5 different species, from sperm whales, dusky dolphins, common dolphins, killer whales, Hector's dolphins. And this presents a huge management issue of how you deal with the data with multiple species in one single area. So that was our sort of approach with discovery. And I will transfer to my colleague, Leszek Karczmarski.
Yeah, this was the approach in discovery, and this chart represents the conceptual framework of discovery, where from field data collection you go through all this pre-matching and matching procedures to all the associated information, sightings info, and survey information with any potential additional information, acoustics, telemetry, genetics, whatever else is being collected during the field work, and then all assembles in this data management component the core part of discovery, and then a number of those outputs and summary of the results. So we'll go step by step very briefly. No, we cannot cover the entire range of components, but the main parts to explain the flow and concepts of discovery. So as Glenn mentioned, different researchers do things differently, and we need to, there is a need for this dynamic system that we can incorporate all these individual preferences and choices. So in this case, the discovery starts from the very beginning with very user specified dynamic systems, where we actually specify all the things you want to have in your system, starting from study area, from you can include your maps, you can choose, or you include the species, you can have multiple species within one database. You go to your individual classification, and you can choose what you will be using for your classification, features you will be using, what type of descriptors. It's a non-index based system. We'll be referring to this a number of times, so it's a number of those classifications aspects are categorical. All the associated citing informations, environmental, group structure, group composition, age, classes, and so on, behavior, and all the miscellaneous informations that actually you define what you want to include there, or anything that will actually be observed during the survey that you cannot predict ahead of time, and it comes, you don't want to get it lost. And since we all have habits, and habits are sometimes good, sometimes not, but they're very, very hard to get rid of. So some people would have certain preferences of putting data in a certain way. Even in simple things like whether it is decimal point or minutes and seconds in geographic position, whether to use original images, whether to use temporary IDs, or perhaps to verify. If you're just a single researcher running with one project with one or couple of catalogs, you might feel that you don't need to verify temporary IDs, although it's probably good to verify them no matter what. But if you work with multiple teams, the verification is critical to avoid mismatches. So all this can be incorporated and is user defined. So going on with this fly chart, all this pre-matching and matching procedures. So first you start with file naming. As it was mentioned yesterday, and with all we know, file naming is pretty critical. You can have all this important information there. This process is automated in Discovery. You can actually choose the whole range of images and have them renamed as a batch process. And you can define how this renaming is going to be structured. In our case, we include temporary ID, date, study area, group size, group number, or encounter, or citing number, depends on how you use it, photographer, and the frame name. But again, this is user-defined. You can decide how your naming structure is going to be. And then it comes all this tedious process of filtering and processing the images, processing, potentially cropping, looking at those parts which are really important, that have all these identifiable characteristics. So if we have an image, like in our case with the work in Africa with elephants, well, you select this part that you really want to, that contains the information. You can get rid of all this background. And actually, you can still improve the image to some degree. We have some tools here that you can increase the contrast, brightness. You can sharpen the edges, and this really helps. Particularly with out at sea, it helps a lot when sometimes just a little bit of extra focus makes those couple of notches more on a dorsal fin a bit more visible. So that's a very nice feature. And then you can plug this into different categories if you like. Quality, for instance, entirely user-defined. There's a number of quality criteria, quality scales. We in Hong Kong use a scale of 1 to 100. Some others use 1 to 6. Some others use basically 3 or 4 or 5 categories, how they're being subdivided. That's your choice. You can create sections like, for instance, mother and calf groups, where you would want to keep a record of all the mom-calf associations and keep a record maternal care on weaning age and potential death records and so on. or we keep all the images. We of course use some to the catalogs, but we don't get rid of any. If they're unmarked individuals, we plug them into the unmarked section. And once you select all the individuals on the images, marked, unmarked, sometimes there will be several individuals on one image, then automatically there is a count going on, how many marked and unmarked individuals are there, so it calculates you the ID ratio, which is very critical in all the population size estimates, which comes very, very handy for those who potentially would forget that this is actually an important thing to keep an eye on. It's not that unusual, surprising. And then matching within a group. Obviously, first within a group, within an encounter, as we call it, where you want to select your best representative images of each group. So you have your query image, and then you compare it to your selected images, basically to your catalog. And you can choose. You can use just one query image, or if you're really good, you can increase your number of query images. You can make the matching to few individuals at the same time, if that's if you know your animals really well. And matching can be done within in horizontal and vertical mode. Depends on what species you work with. For dolphins, dorsal fins, you would use the horizontal mode. Whales are generally, in this case, grey whales are mostly used in a vertical mode. And again, query images and your sample from the catalog to verify the query image. And you can actually you can individually decide how many of the catalog images you want to have shown at the same time. So it could be 6, like for the dolphins. You can choose only use 1 or 2 or 8 or whatever you prefer. So you make this selections at the beginning of in your settings of discovery. And in some cases, you can actually, if you like, you can use this thingy here. English is not my home language. And you can do individual zooming for to pull out some of the specific features of so individual per image, not all of them, or not removing all the others. So, but it is non-indexed method, so that actually your eye is the final apparatus to decide what you deal with, and basically you search through the catalog by filtering and sorting the database. And once you find a new individual, like a newt in this case, from Hong Kong, new individual or match. Well, then you include all the individual information, such as your ID, your species, your group, your date of sight, and so on, your time, if that's important. Your locations. Your location can be imported from the file or entered manually. Individual features, those that never change through the lifetime of the individuals. Obviously, sex generally doesn't. Day of birth doesn't. The age might change, but the day of birth will remain the same if you can estimate it. So that we will be included there. And your user-defined non-indexed classification, as well as all the miscellaneous user-defined informations, such as, for instance, your age class, which will be changing throughout the life history of the individual, your potential mark change, growth. If you work with small enough critters, they can measure how much they increase in their size and so on. So this will be all the pre-matching and matching procedures, and then we go to sighting informations. And our concept was that basically we took our data sheet, and that's just an example of our data sheet from Hong Kong, and we decided that all the information we're collecting, it's obviously important. So we want all this information that we have in a field data sheet to go to the discovery and be freely accessible at any given time. So any information that's there, which will include all this kind of bookkeeping for the survey, all the effort, time, photographic info, and so on, all goes in. All the environmental information, at least at the beginning and the end of the encounter, is in our procedure. All this stuff that comes, like the surveys of, let's see, sea state, depth, location, distance from the shore, habitat, and so on. Then I presume everybody that works with almost any mammals, certainly with marine mammals, you keep record of the group structure, group composition across the survey, which might change. So we would do this regularly through the survey. All this information will go in, along with geographic positions and environmental information collected through the survey, and the behavioral stuff. We would have another data sheet purely for the behavioral data, but in a summary data sheet, it will be at least some records of behavior throughout the survey. And then you just go and enter all this into the sighting information. So your group information, your age classes, number of individuals of individual age classes, your photographic data, what camera you use, who did the photographs, who took the notes, and your locations, again, either imported or manually entered environmental data, any comments that you might have to this specific sighting. You never know what can come up during field work. And this miscellaneous, which in this case, in the example of the miscellaneous, will be the biopsy. Okay, so we took the biopsy, categorical, yes or no. If we did, whom did we sample? Was this the particular individual there? In your other file, when you enter your new individual, you will replace your in this slide just a little bit earlier. You would replace your temporary ID with a new permanent ID. But you can also assign potentially a name if you want to use. If you're better with names for individuals, as I am. I never remember if it is 024, 035, but if it is John or Mary, I will know exactly who it is. So you can then have this information in your miscellaneous info, like the biopsy data, and it sits there forever. Along with all this geographic positions, all this information that we collect throughout the surveys, all the track data that can be incorporated, and goes along with every encounter data. And after this, the main component, the most complex, which again is going to talk about it, data management, which is really the essence of Discovery.
So once you have all your data in, into a central database, you have a lot of procedures you have to go through, and you constantly are checking your database, re-verifying your IDs, and also looking at the individual information. And so you can see this is the catalog of your individuals, which gives you summary data of how many images, how many sightings, when did you first see that individual, when is the last time you've seen that individual. And then when you click on the individual catalog, you can see all the images associated with that individual. So these are the different re-sightings. And you can even have multiple images within a single day. So if you want to include 100 individuals of the same individual within a group, well, sure, go ahead. But usually we only have to include one photo per sighting. But then you can also have an important aspect, I think, is a cross-link between different catalogs. So, for example, this is our catalog for gray whales, but what we call individual 083, another study calls individual 062. So it's a very important thing because they process their IDs, and if we want to make use of all the data, we have the important cross-link between the two studies. And then you have the summary information of your sighting records. And here you can partition your data by your study site if you want, or your species, or the data of study. And this can be, partitioned in any combination of ways. And for example, this is just one date where we pull up the information, and you can see all the sighting data that were recorded. So here's sort of the standard sighting information. And then for any of these sightings, you may have multiple records for each one. So in the environmental, as we're on this group, we had multiple records here. And then, as well as the geographic position. So we just click on a button there, and it pulls up that information. And so we can re-verify that those data have been entered correctly. And then we constantly want to review our database, look at it, and there's this, it's all in Access, so you can use Access if you'd like, or we have this interface here that also lets you visualize all the different tables you have in the database. and it has some QA, QC functions that help you identify potential issues. Like this is a function that goes through and says, oh, you know, you have some problems. You don't have those images on your actual physical disk, so you got to go find those images. So that helps to go through your data and make sure it's all there. So there's all the tables, and then check for directory. And then, if you already have existing data into different processes, as you can do different imports. So here's importing your images into a database, which can use the file name structure. This is sort of a quick import. Then we have a more complex, versatile import, where if you have information in your EXIF part of your data, you can put that as like date and time or geographic position. You can automatically extract it from your EXIF data. Or if you have the information like ID or date as part of your file name, you can put that information into the data and it automatically enters into the system very rapidly. But then a lot of people already have data in different forms. We have our import for discovery databases, which if you have multiple studies that are working with discovery, you can just import them in really quickly. Or if you have your data in a different format in Excel or Access, you can also use this dynamic import, which says that